All right, I think we will go ahead and get started in the interest of time. We welcome everyone to our annual SWOG NCORP research-based clinical trials workshop. This is where we come together to discuss topics related to the conduct of clinical trials in the 5 SWOG NCORP committees. I'm Monica Yee, and I am the Data Management Program Director for Cancer Control and Prevention Trials in Seattle at the Statistics and Data Management Center. In the past 6 years, we've met in person as well as virtually. We hope to be able to see you all soon in person, and we welcome all of you to this virtual session today. Kind of getting to know the controls a little bit here, faster than we thought. There we go. The SWOG NCORP research base supports the research activities and goals of the NCI NCORP in designing and conducting trials in key focus areas, including cancer care delivery. In addition, SWOG is committed to addressing cancer disparities, research questions. So a couple of housekeeping items. Matt, would you like to go over the mute button and other controls on the Zoom?
Hi. So for everyone, all the attendees, when it comes to Q&A, to unmute your mic at the bottom left-hand side of your screen, you'll see a little mic that says mute on it. If there is a line going through that mic, that means your mic is muted, and you'll have to click it to unmute to talk. If you don't want to use your mic or you don't have a mic, over to the right on the bottom of the screen, about halfway in the middle, you'll see a chat box. If you click on that chat button, it'll bring up a window and you can type in your question in there, and all the panelists and presenters will see that as well. And I'll hand it back over to you, Monica.
Okay, great. Thanks, Matt. So a couple of other things. The workshop landing page on swog.org has an agenda. And on the second page of the agenda, if you are seeking SOCRA continuing education credits, there's a signature page on the second page, so be sure to keep that for recertification. Also, we have a list of active and accruing SWOG NCORP clinical trials and a presenter contact list for you. And this session is being recorded in case you would like to refer to it anytime in the future. Today's agenda features 3 presentations related to the area of cancer clinical trials. And we'll receive an update from NCI's perspective on cancer care delivery research, and we'll get an up-to-date activity information about the Recruitment and Retention Committee in regards to addressing cancer disparities. This session will wrap up today with a special panel of individuals you interact with on a day-to-day basis, our clinical trial data coordinators, who will be addressing some topics of interest about data quality. So before launching into the agenda, let's get to know the team behind the SWOG research base. We are a team working together along with all of you to achieve so much more than working alone. The SWOG NCORP research base is guided by the Vice Chair, Dr. Don Hirschman. Drs. Cra and Dr. Lyman serve as executive officers for the NCORP research committees. There are 5 scientific SWOG research committees with hardworking co-chairs for each. These committees promote and conduct trials to support the NCORP's goals to reduce cancer risk, improve cancer outcomes, expand access to care, increase quality and balance cost, and reduce cancer disparities. Our team of protocol coordinators at the SWOG Operations Office in San Antonio oversee the critical role for protocol development and maintenance. I also want to mention that concept and protocol development also involves SWOG's financial, grant, and legal specialists at the group chair's office and the Hope Foundation, who are not pictured but play an important role in concepts and protocols moving forward in this area. Our NCORP-based Biostatistical Leadership resides in Seattle at the SWOG Statistics and Data Management Center, or SDMC for short. Dr. Unger and Vadia will be in this team, will be sharing presentations with us today. All of SWOG's statisticians are key players in trial design and ensure that trial results are complete, accurate, and reliable. Many of you have corresponded with the clinical research data coordinators, who are the liaisons to the study site staff. They also review trial data and participate in trial development. Our data control technicians provide support in data management activities, and we call with the protocol or data question, they're the voice that greets you. In addition to the 5 NCORP research committees, we liaise with 3 SWOG support committees. SWOG's Recruitment and Retention Committee has a focus on diversity in SWOG trials, as well as a focus on general trial recruitment and retention. Dr. Caban Holt, chair of this committee, will provide an update on the committee's activities today. Patient advocates represent the patient voice at committee meetings and trial development, and we are appreciative of their contributions in all stages of the trial life cycle. Last but not least of our research committees, or excuse me, our support committees, is the Oncology Research Professionals, or ORP committee. ORP liaisons provide input and feedback on study procedures and administration of the study at the practice level. So note that there is an open liaison position for the prevention epidemiology. So if you're interested, please let us know. We'd also like to recognize Dr. Parker, SWOG's program officer at the NCI Division of Cancer Prevention, for support and guidance. SWOG NCORP also receives support from so many others at the Division of Cancer Prevention and Cancer Care Delivery, such as Dr. McCaskill-Stevens, Dr. Geiger, Kate Castro, Marge Good, just to name a few, and there are many others. So we've reviewed the who's who of SWOG research. So let's briefly look at the SWOG NCORP trial portfolio, where there are opportunities for you and your patients to be involved. For more information about these trials, please attend the committee meetings in the next few days. And please take note in the participating sites column, notice that most of our trials are open to all sites and not just limited to NCORP sites. There's only one trial limited to NCORP sites only, which is a cancer care delivery trial. In the Cancer Care Delivery Committee, in addition to S1912CD that was newly activated, and we're looking for registrations for our NCORP sites, we also have S1703, which is at 14% accrual, which is a perfect opportunity for any of your sites who might be interested in getting involved. There's plenty of time to pick up that trial and help us reach our goal in that area. Cancer Survivorship has an important trial to study the prevention of cardiac dysfunction in the subset of breast cancer patients, and it offers an excellent opportunity with only 31% accrual at this time. In Palliative and End-of-Life Care Committee, S1820 is past its halfway point of accrual, so if we can work together to complete accrual for one of the first palliative care and trials in SWOG, that would be amazing. The Prevention and Epidemiology Committee has 3 trials open for participation, 2 of which activated in 2020 and 1 in 2013. Please review them and see if your site is able to participate. There are 4 trials in symptom control and quality of life open to all sites. S2013 and S1614 are newly activated and reactivated. There is a study meeting tomorrow, so please join that to learn a little bit more about those studies and to hear from the study chairs. We'd also like to congratulate all of you for quickly accruing to S1714. We are at 95% accrual, so we anticipate that this goal will be met very soon. So with that, let's get started with our agenda. Our first presenter is someone you may be familiar with because of SWOG research publications, such as Quantifying Years of Life Saved by federal-sponsored trials, publications on socioeconomics and access to care and cancer disparities have made international headlines. It's my pleasure to introduce Joe Unger of the Statistics and Data Management Center in Seattle, who will present on the impact of the COVID-19 pandemic on cancer clinical trial participation and new study activations.
Thank you, Monica. Can you hear me?
Yes.
So thanks for that introduction. Matt, maybe you could, if you wouldn't mind just advancing the slides for me, it'd just be easier. So thank you. I'm Joe Unger from the Fred Hutchinson Cancer Research Center and a senior statistician and health services researcher with SWOG. As noted, I'm going to discuss the impact of the COVID-19 pandemic on cancer clinical trial activations and new study registrations and new study activations. Next slide, please. So, as we all know, the COVID-19 pandemic has caused severe interruptions in care for patients with non-COVID-19 diseases. As research into COVID-19 treatments has dramatically increased, a key question is whether the landscape of clinical research for non-COVID-19 diseases has also changed, especially in the US, where there's been a disproportionate number of cases and deaths from COVID-19, especially early on in the pandemic. Next slide. Why is this important? Well, because advances in new treatments for patients rely on the conduct of clinical trials, which represent a key final step in proving the efficacy of new therapies. So interruptions in the conduct of clinical trial research could slow the development of new treatments for common illnesses, especially cancer. Next slide. So today I want to focus on evidence about the impact of the COVID-19 pandemic on 4 different evidence domains. Starting from the left, the pandemic has impacted the launch of new trial activations, patient willingness to participate in trials, the enrollment to active trials, and potentially disparities in trial enrollment. Moreover, these domains can influence each other as suggested by the light gray arrows. For instance, patient willingness to participate in trials will certainly impact enrollment to active trials and may exacerbate existing disparities in access to trials. Next slide. So with respect to new clinical trial activations, my colleague, Dr. Zhao, and I examined the association of the COVID-19 outbreak with new clinical trial activations using data from clinicaltrials.gov. We included all interventional and observational oncology, cardiovascular, and mental health studies over a nearly 6-year time period using interrupted time series Poisson regression. Next slide. In total, we examined more than 62,000 trial activations. We found that during the initial COVID-19 outbreak from February to May of 2020, monthly trial activations dropped 43% for US-based studies and 23% for non-US-based studies. So it was notably larger for US-based studies with a relative risk ratio of 0.73. During the initial reopening phase from June to September of 2020, a rebound in new study activations was observed. Fortunately, however, the rebound was weaker for US-based studies compared to non-US-based studies with a relative risk ratio of 0.87. Next slide. The findings in oncology in particular are shown in this table. During the initial pandemic period in the US, 197 studies were expected to be activated based on prior trends, but only 121 were actually activated, a reduction of 76 studies or 39%. In contrast, for non-US based studies, the relative reduction was only 20%, and similar trends were observed for cardiovascular and mental health studies. Next slide. So these findings are consistent with the disproportionate burden of COVID-19 in terms of diagnoses and deaths during the initial phase of the pandemic in the US. So an important question coming out of this is whether subsequent outbreaks have had a similarly negative impact on the initiation of new clinical research studies for non-COVID-19 diseases, which is something we're continuing to monitor. Next slide. A recent study also examined patient willingness to participate in cancer clinical trials during the pandemic. We surveyed with my colleagues at the American Cancer Society, survivors participating in the American Cancer Society Cancer Action Network Survival Views panel. To participate, panelists were 18 years or older US residents and had been diagnosed with and or treated for cancer within the past 5 years. And the benefit of using this panel was that the designers at the American Cancer Society were able to move very quickly and ask a series of questions regarding disposition toward trials during the early stages of the pandemic. Next slide. We found that among 933 respondents, nearly 3/4 were female, and 80% indicated the pandemic made them no more or less likely to participate in the trial, which was a good thing. However, among the remaining respondents, more than 7 times more were likely to indicate that the pandemic made them feel less likely to enroll in a clinical trial, with percentages of 18.1% versus 2.4%. Response patterns were similar across these various demographic, socioeconomic, and care settings as shown in the figure. You can see that regardless of patient group, many more patients indicated, or survivors indicated they were less likely to participate than more likely to participate. Next slide. Moreover, among the 164 respondents reporting that they would be less likely to enroll, the most common reasons given were fear of increased COVID-19 exposure at about 70%, or difficulty accessing care during the pandemic, which was reported among 18%. Next slide. So in summary, nearly 1/5 of the patients with cancer reported that they would be less likely to participate in a trial because of fears surrounding COVID. These findings were consistently observed across evaluable demographic and socioeconomic patient groups. And they suggest that as long as high rates of COVID-19 cases exist, patients with cancer will be hesitant to participate, more likely to be hesitant to participate, even when sites return to pre-pandemic status. Next slide. So fairly early on in the outbreak, we also examined how the initial outbreak may have influenced enrollment to SWOG trials. We noted there was a clear correspondence between increasing cases in the US, highlighted in the blue, and decreasing enrollment in SWOG trials in aggregate, highlighted in the darker color with, I guess, black. And so you can see that the cumulative COVID-19 cases in the US exploded beginning in March of 2020. And then there's that corresponding very steep drop in weekly aggregate enrollments to SWOG trials. And it turns out that this observation was consistent across the network groups, as reported to Dr. Dorshout to NCI leadership there. Next slide. Then a year into the pandemic, we followed up by examining the full 1-year experience of cancer clinical trial enrollment, including whether patterns of enrollments differed over time in conjunction with COVID-19 mortality waves, and whether existing inequities in trial participation may have been exacerbated. Next slide. So we examined weekly initial enrollments to SWOG clinical trials through February of 2021, looking at both treatment and cancer control and prevention trials altogether and separately. Again, we used an interrupted time series analysis, and we examined pre-specified intervals of interest, including the initial COVID-19 wave, the initial recovery period, and then the very severe winter 2020-2021 wave. Next slide. So among the nearly 30,000 patients that we looked at, we found a 9% weekly reduction in enrollments compared to expected rates during the initial COVID-19 wave. And now this doesn't sound very much, but these are compounding effects every week, so they accelerate very quickly. Enrollments recovered at a rate of 4% a week during the initial recovery period, but then dropped again during the very severe winter 2020-2021 wave. Although it's worth noting that the drop during the severe 2020-2021 winter wave was much more mild at only about 2% per week. And you can see in the figure the patterns of decreasing and increasing enrollments in the top panel track very closely with the COVID-19 waves shown in the bottom panel. Next slide. So overall, during the entire first year of the pandemic, there was a 23% relative reduction in enrollments to all trials combined. Although this differed very substantially by the type of trial. For treatment trials, we found only a 9% overall reduction compared to what was expected, whereas for cancer control and prevention trials, we found a 46% relative reduction compared to what was expected over the entirety of that first year. Next slide. And then this figure shows relative change in enrollment during the pandemic over the entirety of that first year within patient groups. During the entire first year of the pandemic, we saw consistently large reductions in enrollments to cancer control and prevention trials for all demographic subgroups, whereas reductions were consistently more modest across groups for treatment trials. Of note, for both treatment and cancer control and prevention trials, there were proportionally fewer patients enrolled during the pandemic from states with higher COVID-19 related excess death rates, whereas for states with lower COVID-19 related excess death rates were similar. Next slide. So in conclusion, the COVID-19 pandemic has had an immediate and devastating impact on the conduct of cancer clinical research. It resulted in the delay or cancellation of new trial activations. It made patients more fearful of participating in trials, and it led to dramatic drops in enrollment during the initial wave of the pandemic. Next slide. But on the positive side, there was also evidence that the clinical trial system rapidly adapted to the circumstances of the pandemic. New guidances put out by the NCI and the FDA provided flexibility in trial conduct, as many of you know, with regard to strategies such as remote consent, virtual visits, local healthcare practitioners being allowed to administer protocol therapy, as well as the allowance of alternative labs and imaging centers to conduct protocol required tests. And likely as a result, trial participation rebounded and subsequent drops were not nearly as severe as suggested by the initial pandemic wave. Next slide. In fact, in SWOG, overall treatment trial participation during the first year of the pandemic did not, surprisingly, substantially diminish compared to what was expected. Although this may have come at the expense of enrollment to cancer control and prevention trials, and could, and very well may likely, reflect institutional prioritization of resources to target the conduct of trials that were most directly related to patient care. Next slide. So in this sense, the pandemic challenged the trial system to adopt strategies that had long been discussed. In that way, it was sort of functioned as a kind of natural experiment. Efforts are ongoing to evaluate whether data quality suffered during the pandemic. If not, these new strategies could be adopted as routine practice in trials, potentially benefiting the conduct of trials and patients interested in participating in trials for their care in years to come. Next slide. And I just want to acknowledge my collaborators and especially all of the SWOG researchers and investigators on the front lines of patient care during what has been clearly an extraordinary time. Thank you.
Thank you very much, Joe. If anyone has any questions, we have a couple of minutes. If there are any, I don't see any questions that have come into chat. If you have any questions, you can enter them in chat.
I see a question from Katie Arnold, who asked how well clinicaltrials.gov represents international studies. I think it represents them fairly well. Regardless, it's the relative reductions we were comparing in any event. So even if there was a sort of an aggregate underrepresentation, that relative reduction allows a fair comparison, more or less.
Great, thank you, Joe. I don't see any other chat. Do you? Were any addressed directly to you?
Nope.
Okay.
Give back my time.
Well, thank you so much for your presentation and for sharing your research with us. If anyone has any additional questions that you can think of, go ahead and put those in the chat and we can forward those to Joe and respond to you after the workshop. So our next presenter is Dr. Ria Vaidya. Our patient-reported outcomes, or PROs, are a very important component of many SWOG trials. So Dr. Vaidya, who's a staff scientist with expertise in analysis of patient outcomes, will walk us through PROs from principles to practice.
Thank you, Monica. Hi, everyone. My name is Ria Vaidya. I'm a staff scientist at the Fred Hutchinson Cancer Research Center and a statistician at SWOG. I also coordinate SWOG's patient-reported outcomes core. So what I'm going to talk about today is beginning with a brief overview of PROs and how we use them in SWOG studies. Next, I'll go on to describing some of the principles that guide PRO design and analysis in SWOG trials, and mainly how they translate into practice at the site level. And finally, I'll close with a bit of a brief overview of electronic patient-reported outcome data collection and how that ties in with some of these principles and practices that we talk about. So I'm sure many of you are familiar with this from working on SWOG clinical trials, but just to briefly touch on it, what are patient-reported outcomes? So PROs are essentially a report of the status of a patient's health, and this report is coming directly from the patient without any interpretation from their clinician. So PROs do include quality of life, but that's not the only thing. They can also include other things like functional status or behaviors, patient diaries, things like that. So quality of life and patient-reported outcomes really can be thought of as overlapping concepts. So why do we collect PROs in our clinical trials? Cancer treatment can impact several aspects of patients' lives. And what PROs do is they help bring in the patient perspective through a direct report of their well-being, their symptoms, how they're feeling, their functional status. And along with clinical outcomes, PROs can help provide a sort of more complete picture so that the patient and their clinician can make a treatment decision together. So for example, if you have treatments A and B, that are similar, but we have PRO data on symptoms and toxicities, that can help us determine whether treatment A or B might be better for the patient based on that additional PRO data. It can also help identify needs and concerns for cancer survivors, things such as rehabilitation, etc. So how do we use PROs in SWOG studies? So in many of our NCORP studies, PROs are often the primary outcomes. So to give you a few examples, for S1200 and S1202, the brief pain inventory was the primary outcome for these studies. For the recent study, the ongoing trial S1820, the MSKCC bowel function instrument is what the primary outcome is based on. But it's not just in NCORP studies that we use PROs. They're also widely used as secondary objectives for many of our treatment studies. And here are just a few examples of ongoing treatment trials where we're using PROs. And while we don't use PROs in every single treatment trial in SWOG, we do have a process for their inclusion. It begins with the investigators developing a PRO proposal within their disease committees. The proposal then comes to the PRO core for review, where it's reviewed for feasibility, for budget, and recommendations are made for how to develop the PRO study further. After that, it goes through the SWOG executive triage review process, and once that's approved there, it finally goes on to DCP, where if it is approved, the PROs will be included in the protocol and all of the other process of provision of site credits happens. For NCORP trials, because as I mentioned, our PROs are often primary endpoints, we don't necessarily follow the same process, but the PRO core does review what PROs are going into NCORP studies. And there is the SWOG triage review process and the DCP review process that we follow for all trials. So now I'm going to talk about some of the guiding principles that guide the design and analysis of PROs in our trials. So on the design side, it begins with having PRO-related research questions that are driven by specific hypotheses. Second, we use validated instruments as far as possible. Thirdly, it's important to assess the feasibility of inclusion of PROs in the context of the overall, the clinical trial, the parent trial, and ensure that inclusion of PROs will not interfere with the conduct of the main parent trial. We always want to limit patient and site burden in terms of what PROs we're assessing. And we want to avoid assessment bias, specifically in terms of how our PRO assessments are timed. On the analysis side, the principles we think about are what sort of statistical design are we using so that these studies can be easily interpreted clinically, and we have missing data strategies both on the front end, how do we design the study so that we don't have a lot of missing data issues, but also when we're analyzing, how are we going to review for missing data in our studies. And all of these principles, but especially the design principles, are really interconnected with each other, and not following the principle on one of these principles can kind of have a cascade effect. So if, for example, if there aren't hypothesis-driven research questions for PROs, it can end up with a lot of patient and site burden because the study may end up using a lot of instruments because of the lack of a specific hypothesis. So it's really important to think of all of these sort of together as a concept. And when we have all of these PRO guiding principles, the goal ultimately here is to have timely data collection and accurate data so that we can generate PRO findings that are easy to interpret. And our study protocols are designed based on these guiding principles. So what I'm going to do over the next few slides is just talk a little bit about how sites can help us adhere to these principles in the data collection process, both overall and with respect to some specific principles that I discussed on the previous slide. And site staff play a really important role here because they are a link between patients and the stat center. And sites are really critical partners in ensuring that we have good quality PRO data on which our findings will be based. So let me begin with just a few overall sort of practices. So before registration, it is important for any site staff who are involved in PRO data collection to complete SWOG's PRO questionnaires training program. It is available on SWOG.org. If you go to Clinicaltrials and the Protocol Workbench section, there is a link to the training there. I believe the training is also in the process of being uploaded to the SWOG learning management system, so it will appear there as well. And the link that's mentioned, that I just mentioned on SWOG.org, I believe there was a memorandum that went out to sites at the beginning of 2021, also sharing that link. So that is the main interim training that we're using right now. The other things to do before registering patients are reviewing the study protocol for all of the details of the PRO assessments, how many forms there are, what the time points are, instructions for completion, data submission instructions, all of those things. And also to prepare to answer any patient questions about PROs in the study. And a lot of that information can be found both in the protocol and in the consent form. So now let me talk about some of the specific principles and sort of what are some practices to follow to ensure that we're adhering to those principles. The first one I want to talk about is validated instruments. So when we talk about validated PRO instruments, these are instruments that have been through a rigorous testing process to ensure that they are actually measuring the concept that we're interested in measuring. So it's very important that when an instrument is validated, that it is presented to patients as it is intended to be. So, some of the things to do here are checking if all of the pages are correctly printed so that there isn't a block of questions that goes missing because a page wasn't correctly printed or didn't align properly. And also to ensure that the patients really understand all of the instructions for completing the questionnaires. Like, this is a great time to answer any questions from them and make sure they have a full understanding of what it is that they are expected to do. Next is avoiding bias, and I've kind of divided this into two chunks here. So first I'll talk about it in terms of avoiding bias in terms of the timing of questionnaires. So here it is very important to follow assessment times or windows for completing questionnaires as is indicated in the protocol. Scheduled assessments should be continued as indicated, even if the patients discontinue protocol therapy, and those instructions are laid out in the protocols. And the third would be following protocol instructions for PRO assessment if the patient misses a clinic appointment. The next section of avoiding bias, and this is very important, is about the actual responses to the PRO questionnaires. So it's very important for PROs that the patients answer the questions themselves without any influence or help from others. So others here could be a relative or a friend who has accompanied the patient on their clinic visit. It might be clinicians, it might be site staff. So anyone really. The whole point here is that the patient answers the question for themselves without anyone sort of influencing what response option they should select. Now, it is very much possible that a patient might need assistance with reading questions if they forget their eyeglasses, for example, and they can't read the forms themselves. In those situations, it is perfectly fine to read the questions out to the patient, but while doing so, it is very important to maintain a neutral tone so that there is no appearance that the patient is expected to answer the question in a certain way. The next critical component we'll talk about is missing data. So in any study, when we collect a lot of patient data, despite our best quality control procedures and despite sort of how we design studies, it is likely that we will have some sort of missing data or data issues. But it's definitely possible to take steps to ensure that we minimize that as much as possible. So a few steps here for things that can be done with respect to missing data. So the first is reviewing the questionnaires after patients complete them. So the review here really is focused on whether patients have completed all of the questions that they are supposed to complete on the form. And this is the time to offer the patient an opportunity to answer any questions that they may have missed. And it may so happen that the patient intentionally did not want to answer that question, and that is fine, but this is just a chance to make sure that it's not just that they missed the question as opposed to they didn't want to answer it. This is also an opportunity to potentially correct the patient if they have multiple responses selected for a question where they only are supposed to select one. This is a good time to ask them if there is one response they want to select out of the more than one that they have selected. For missed assessments, it's still important to submit the PRO cover sheets and indicate the reason for non-completion of the forms. And finally, when the data from these paper forms that patients have completed is being entered into RAVE, it is very important and very helpful to DCs if you can review for errors and catch any of them before the data are actually submitted. So another strategy to deal with missing data is electronic PRO data collection. And this is something that has been in the works for quite a while. You may have seen it in studies done by other groups, and we've started using Epro in SWOG studies as well. So what Epro is it's just any means of electronic capture of patient-reported data. It can be mobile apps, it can be a web-based application, links, things like that. And the reason to use Epro is there is this ability for patients to complete the PRO forms at home. It reduces the burden of paper forms and data entry for patients, and it also improves data accuracy and compliance. So just to briefly go over some of the logistics of Epro in SWOG studies. So NCI-sponsored studies use Metadata's Patient Cloud app for PRO data collection. These can be used on the patient's own smartphone or tablet that has an internet connection, or they can be used on site-provided devices. And when a patient enters data into the Patient Cloud app, it gets directly uploaded to RAVE. So site staff don't have to separately enter that data into RAVE. That upload is just automatic. And as far as instructions for access for site staff and learning materials and training materials related to ePro, those are all available to site staff via iMetadata. And all of the instructions are in the study protocol as well. Currently, we're only using ePro in S2013, the study that recently opened. So a few ePro considerations for site staff. So one thing might be to check in RAVE if the patient has completed ePro assessments. If they have completed this visit, might be a good time to encourage them to keep completing future assessments, or if they've missed assessments, this would be a good time to remind them to complete their ePro assessments. And this is, of course, assuming that the patient has chosen to use ePro for completing their PRO assessments. Other things include following any protocol instructions on PRO cover sheets for patients who are using ePro. And our data coordinators are monitoring all questions, and especially ePro related questions that come in from sites. So if you have any question, please email them to cancercontrolquestion@crab.org. And any ePro related questions will be monitored there, responded to, and DCs will create documentation in response to the questions that come in as needed. So in summary, along with clinical data, PROs help patients and physicians make treatment decisions, and site staff play a really key role in ensuring that we have high quality PRO data for our studies. Using practices based on PRO guiding principles can help us ensure that our PRO studies are successful. I would like to thank all of the members of SWOG's PRO Core, and I would also like to take a moment here to thank Lisa Hansen and Jamie Myers, who have done PRO trainings over the years. And all of the trainings have been really helpful resources in putting this talk together. Thank you.
Thank you so much, Ria. I don't see any questions coming in the general chat. Ria, have you received any directly from anyone?
No, I don't
Okay.
see anything.
All right. Well, thank you so much. And again, if you have any questions, go ahead and enter those in the chat and we can have Ria respond to those after the workshop. With that, I would like to introduce our next presenter, Ms. Ellen Richmond, a board-certified gerontologic nurse practitioner. She's a program director and nurse consultant in the Gastrointestinal and Other Cancers Research Group in NCI's Division of Cancer Prevention. Ellen will be describing the Accrual Quality Improvement Program, AQUIP, which is a dynamic clinical trial accrual improvement program for early phase chemoprevention research.
Okay, good afternoon, everyone. Nice to know that you're out there. Wish I could see you though. I want to first thank you, Monica, for this opportunity to talk about the Accrual Quality Improvement Program, or AQUIP. Next slide, please. So what I plan to cover today is, of course, first I'll give some background and context for AQUIP, and then I'll describe the purpose of the initiative. Then I'll give an overview of 3 of the 6 or 7 major components of the program, namely the multi-scale data collection, accrual zone monitoring plan, and the feasibility assessment scoring tool, or FAST. And then I'll share some of the preliminary results, some of the data that have been collected, followed by a very brief summary. So I'm going to talk fast. Next slide. While most people are familiar with DCP's role in NCORP and large phase 3 cancer prevention trials, DCP actually supports all phases of prevention drug development. And the early phase clinical trials program is a group of 5 multi-institutional clinical networks that conduct phase 0, 1, and 2 clinical trials to hopefully qualify agents for further clinical development through NCT and NCORP and other sponsors. Next slide. So to address the challenge of accrual to these early phase prevention trials, we initiated sort of a deep dive continuous quality improvement approach to data collection and monitoring, and to aid decision-making and study design. And we developed this suite of interrelated tools and resources. The idea is that developing a better understanding of the dynamics of accrual to ongoing trials will help us to improve not only the execution and design of these trials in real time, but then also, of course, apply our findings to future trials. And we found that each phase of the CQI process is integral to the program. And we require a written, detailed study and site-specific recruitment plan and comprehensive documentation of the plan implementation and outcomes. And then we use the data, study it, and every month, both within DCP's review group and with the study sites. Next slide. So more about the multi-scale data collection. Next slide. One important tenet of CQI is that the people who are in the best position to recognize opportunities for improvement are those people in the front lines. So we ask the accrual site staff to collect data throughout the course of an individual's participant recruitment and enrollment process. The data fields, some of which are not routinely collected in screening logs, include the methods employed at every phase of contact and invitation to each potential participant. And when an individual doesn't complete the enrollment process, the specific reason for discontinuing is captured. And then these data also are reviewed monthly to uncover challenging study issues and to possibly inform protocol modifications or new strategies to offset the effects of a study design problem that hadn't been anticipated. In one case, for example, a review of the barriers that were reported revealed that staff had actually misunderstood the protocol, and so more training was needed. And in another, the purchase of home blood pressure kits to decrease the number of study visits made a huge difference. So we really try to stay on top of it throughout the study and at each site. And then we compile the data to for future validation of our equipped feasibility assessment scoring tool, the FAST tool. Next slide, please. So based on that data collection, next slide. This is a cumulative look at the successful strategies used in this program in 2020. And you can see the strategy that was oops, sorry, one back, please. At the top, you can see that the strategy that was overwhelmingly most successful was the use of existing clinic schedules by investigators and clinic staff. And this really reveals the lack of the use of other strategies, especially we noticed social media. Next slide to the barriers. So the barriers, here we show a cumulative look at the top reasons that participants didn't end up starting study interventions, and discounting those who actively or passively declined without providing a reason. The primary barriers are, not surprisingly, eligibility criteria, prohibited concomitant medications, comorbidities, agent toxicity, surgery timing, which in the case of prevention studies is usually the study design called pre-surgical or window of opportunity studies, and of course study burden issues. So the take home here is that the barriers that are in red and indicated with arrows substantiate the basis really for the FAST feasibility assessment tool. Next slide. So next, the other more unusual data set that we collect includes the study and site level events. As you know, some periods of slow accrual are related to, if not caused by, unanticipated conditions, you know, like the pandemic, for example, that occur during the life of the study at any particular site or at a broader level. The pandemic is a great example, but actually over the years, I'm sure you'll all agree that things come up. It can be things that cause negative, have a negative result, and some that are positive. But examples are, you know, at sites, staff turnover, all kinds of staff issues come up. That can take a month or two or three away from study accrual goals. And at the site level, you can have bigger issues like even in the beginning, the staggered opening of the sites decreases the rate of the study overall. Next slide. So next, to investigate the impact of these situations, the data are collected in what we call the equipped journal, and are mapped on this annotated timeline report to provide a visualization of potential drivers of accrual rate changes and slow accrual. And basically it's tells the life story of the study and gives documented evidence of the need for allowances for unforeseeable delays when projecting accrual rates. We call these the predictable unpredictables. But basically these, next slide, these events become the war stories that some of us frustrated clinical trial people sit around and talk about at the end of meeting days like this one. So about the accrual zones, we also wanted to approach the issue of languishing studies more systematically with milestones and with, importantly, with frequent monitoring schedules. And for this, we looked to other disciplines and other clinical trials programs, and we modified an accrual performance zone system that the National Heart, Lung, and Blood Institute uses for accrual oversight of very large grants. Next slide. So at the bottom of this slide, you are seeing an accrual zone monitoring report. These are designed to ensure investigator compliance and accountability, and to keep timely accrual front and center in the minds of study teams. We provide this visualization of the projected accrual timeline with milestones, and the chart compares the actual accrual to those milestones on a monthly basis. And these, you can see these are presented on a background of color-coded accrual performance zones that have corresponding corrective actions, which are determined by how well or poorly accrual is going and why. It also provides reports of and visualizations of the rest of the participant level data that I just showed you. And again, the data are reviewed on a monthly basis and by both DCP staff and leaders of the program at the upper levels, and at each study's monthly calls with the study sites. Next slide. So the next slide shows the guidelines for corrective action and for assessment of study viability, which are based on the level of achievement in relation to the projected accrual at a given time point. Sometimes these are known as stopping rules, not very often, but sometimes they're stopping rules. So you can see in the right most green cell, there were people dancing in that cell to show that when you're in the green zone and you've approved 90% of the projected accrual, we're happy to celebrate the success. But if accrual fails and falls behind, depending on the zone and how far behind accrual is, corrective actions are required. And they range from adding new strategies to increase up to more frequent monitoring to review by DCP leadership for consideration for study closure. Next slide. And after the next slide, I will talk about the third major component of the program, the feasibility assessment scoring tool. This was developed to tackle the problem of unrealistic study accrual rate predictions. Next slide, please. And to minimize favorable or unfavorable study designs. Can I see slide 15 now, please? Are we stuck, Matt?
Ellen,
Yeah.
Matt mentioned there's a slight technical issue
Okay.
at the moment.
Am I in control now?
Oh.
I didn't do it.
Oh, no, I think Matt did. He said he had to back out.
Okay. Well, I can tell you a little bit without the slide, and that is that the idea behind this feasibility assessment scoring tool is that by evaluating the accrual potential of a proposed study based on study attributes, study characteristics, before it's approved, appropriate changes can be made to the study design and proposed accrual rates can be adjusted, or strategies to counterbalance the study vulnerabilities may be developed. And the assessment can justify resource allocation for staffing or recruitment strategies. If changes can't be made and accrual doesn't seem feasible, then you have a basis for a disapproved or resubmitted concept, or more recommendations by DCP staff as to the accrual rate. And of course, these evaluations also can reveal a very highly recruitable, well-written, highly recruitable study based on that FIS score. And this tool is meant for use by both reviewers and by PIs and site staff to enable fair study assessment. Wait till you see it, you'll love it. And reviews.
I think Matt is reloading the PowerPoint presentation right now. So sorry, Ellen.
That's okay. But
I can't wait. I can't wait to see that image though. I can't wait to love it.
Well, just to tell you a little bit more about it, I don't want to take up too much time. The tool, the way that we developed the tool, at least, it was really developed as a pilot. And as a starting point, we based on a consensus among the team members and actually from, based on the literature, we selected 6 cancer prevention design factors as those most likely to influence accrual performance. Next slide. Keep going. One more. Okay, so here you can see in the first column, the characteristics are listed. We have cohort cancer risk as an indication of motivation of the cohort. We have agent toxicity, which is a documented recruitment deterrent, especially in the prevention populations. Then common concomitant medication exclusion, which we know to be very significant in our studies because many of the drugs tested are for prevention are repurposed and very commonly used drugs that have, because they have proven safety records. Then we have two indicators of participant study burden, which are the number of visits and the type of invasive procedures. And the sixth study characteristic I mentioned earlier is the pre-surgical window, which is commonly used, a commonly used study design in solid tumor cancer prevention. And this has historically been very difficult because of patients' reluctance to delay surgery. So then we identified 5 representative instances of each characteristic with respect to their relative effect on accrual and assigned a corresponding FAST score. And that would be between 1 and 5, with 1 being the least conducive to enrollment and 5 being the most conducive. And those examples or instances were then used to determine the scores of all active early phase prevention trials. And then because the study characteristics are impactful individually and in combination, the studies were given a final score based on the sum of those 6 characteristics. Again, the idea is the higher the score, the more likely a trial would be to accrue on time. And the reason for the color coding is because of a hypothesis that the scores should correspond with the equipped zones. Next slide. The next slide is an example of how we would score a study. It's slide 17. There we go. So yeah, this is an example of an ulcerative colitis, a study in an ulcerative colitis cohort testing erlotinib, which is a cytotoxic drug. The study excludes the use of a common drug like protein pump inhibitors and requires 3 clinic visits, 1 non-standard of care colonoscopy, and does not use a pre-surgical window design. And this adds up to 17, so that the equipped FAST score is 17. And based on that, you would, basically that's a relatively low score, and you would assume that, assuming, I guess, first, that you can't change any of the factors because you may sabotage the scientific or clinical basis of the study. In this case, we would recommend an intensive recruitment plan and a conservative monthly accrual rate. Just an example, next slide. So this is basically a list of the 39 studies performed by the early phase prevention networks, with each row being a study and each column representing a study characteristic, with the last column being the study's total FAST score. And they're listed from highest to lowest scores. And I like this heat map type visualization approach because you can tell just by eyeballing it that the lower scores are in the red zone and the higher scores on the top half are, well, there are fewer red zones. So you can see that the threshold really is about 20. More studies that have FAST scores greater than 20 accrued closer to the time proposed. And you can also see, though, that some of the high FAST score studies did not accrue on time. And a couple of studies that scored low actually accrued efficiently. So the FAST score, we realize, is not a guarantee for success because of the random events like pandemics that tend to intervene. I say like pandemics, but we all know there are lots of other things that have happened over all of the years that we've done trials that have a big impact. But this is where the journal data come in. That is, we can often infer the cause of discrepancies between the FAST scores and their predictions based on the study journal events. Next slide. I'll give you some examples. So, for example, the study at the bottom, which you can't see, is a score of 17, but it actually was in the green zone, we would have expected it to have slow accrual, but looking closely, we saw that this study had a group of very well experienced and and PIs with great track records in accrual, and the cohort was highly motivated. And then if you look in the third row, the study that scored 22 but ended up in the red zone. In that case, the lead PI moved to another institution just after the study was approved, and there were also major subcontract delays. And finally, the reason that quite a few studies with high scores don't meet their projections is just that their projected accrual rate is too high and unrealistic. So our conclusion is that studies often don't accrue as predicted, either because of unforeseen events, very common in the life of the study, or because of unrealistic accrual rate projections. Next slide. Next slide. Actually, let's skip this slide. And yeah, just to summarize, what we're pushing here with EQUIP is accrual conscious study design, which is really another way of saying patient conscious study or participant conscious study design, because we want to be able to accrue patients and we want them to like it. And we found that the study and site-specific recruitment plans were people with realistic predicted accrual rates are very important, and we collect a lot of data and use those data, monitor frequently, and we found that the EQUIP feasibility assessment scoring tool has been useful, and we hope that it can be adapted for other settings. Next slide. Next slide, I think, is where I thank everyone. Oh no, this is the EQUIP webpage, the current EQUIP webpage, if you want to check it out, dcpequip.com. But I would like to thank the EQUIP project team, which includes nurse colleagues, Maggie House, Ann Ryan, Eileen Diamond, and Lisa Benson, and all of the members of my group that put up with me talking about EQUIP all the time. So thank you very much, and I'd be happy to take questions.
Thank you so much, Ellen. This is really, really very interesting, and the website is so robust. So although SWOG doesn't currently have early phase chemo prevention trials on our menu, the website has all of these useful resources for trial administration, such as overcoming clinical trial barriers, reaching different target audiences, and recruiting diverse populations. And what I really was interested in was this very robust 130-page toolkit on the website with so many useful resources that may be helpful for administration of any of our trials that we can adapt, like you mentioned. So thank you very much for this presentation, and I urge everyone to check it out on the web. If you have not yet, check out their website. Very interesting, and forward any questions that you might have to Ellen. Thank you again.
Thank you.
Our next presenter is Kate Castro, who is a nurse consultant in the Office of the Associate Director of the Healthcare Delivery Research Program. As the CCDR Program Director in NCORP, Kate works with research bases and sites to identify accrual challenges and strategies to address these challenges at the individual and practice level. So today, Kate will provide an overview of the overall cancer care delivery portfolio and how SWOG studies align.
Great, thanks, Monica. And thank you for always considering us in the cancer care delivery portfolio as you plan your event each year. We used to say we were a brand new area of science. Now I think we're now just new, if we can say that, or we're not brand new. And a lot of you are really getting to know our work a lot better. So it's really a pleasure to always be here and share some updates on our portfolio. So that being said, as we kick off here, I just wanted to share with you sort of where we're at with the portfolio. It doesn't seem to be advancing that. No, there we go. So we have kind of some new updates that have come out in the last couple of months. And the first one at the very top on our recently activated list is a SWOG study. And I know you're going to hear more about it during the meeting this week, but I wanted to share to make sure everyone was aware of the credit study, which is S1912CD. This is a study looking at financial hardship through the delivery of a proactive financial navigation intervention to both the patient and their spouse. So the dyad is enrolled in the study. And this is really exciting for us because many of you participated in the S1417CD study, which was an observational study that was done in metastatic colon cancer patients. ECOG Akron also had an observational study in adjuvant colon cancer patients. And then there was some work done within the Alliance research base looking at financial hardship for patients who have CLL and other hematologic malignancies. So seeing that work in the observational area now move into this interventional work is really exciting for us. And this is a patient level randomization. It's activated and I know they're just ramping up to get started with enrollment. So please, if you're interested in participating, reach out to the SWOG teams here and you'll get a lot more information about this tomorrow at the CCBR committee session as well. We have a second study that is approved and awaiting activation at the Children's Oncology Group, and that's looking at financial distress during treatment for pediatric patients who have acute leukemias. And the participant on this study is the parent of the child who has ALL, and is understanding what that parental and family unit experience when a patient has leukemia, and then what are the financial ramifications down the road for the parents. And this study is just awaiting some translations to be done for its documents, and then it will be moving forward with activation. So we hope by the end of the calendar year, you'll be seeing that. And then recently approved at the CCBR steering committee is another SWOG study, which is the S2108CD. And it's a cluster randomized trial comparing educationally enhanced genomic tumor board compared to usual practice. Again, you'll be hearing a lot more about that tomorrow at the CCBR subcommittee session, but really excited about this work coming forward as well, because this is a whole new area for us. And I think this is a way to really reach some of the groups that don't have access to genomic tumor boards, to see what are some of the strategies that we can get this implemented. I know there's been a lot of excitement about that at previous meetings. One thing I do want to note is you see the asterisks there regarding CCBR protocols, and Monica did a beautiful job of saying this up front as well, that cancer care delivery research work, when you see a CD in the numbering convention of a study, that means the work can only be done if you are an NCORP, either a sub-affiliate or an affiliate. And this is because of the funding mechanism. The NCTN does not have cancer care delivery funding mechanisms. So while we would love to have everybody be able to participate, there are limitations on the funding that we have. So all of the CCBR protocols are limited to just the NCORP affiliates and sub-affiliates if there is a CD in their numbering convention. Now moving on to some of our other work that we have in our portfolio, we have 9 protocols open and accruing. I'm not going to go through all 9 of those now. They're certainly posted on the CTSU, but wanted to highlight some of them that have really come out of the gate and doing a great job this year. That is the SHARE study from the Alliance Research Base, the 231901 CD, and that's improving patient-centered communication for breast cancer patients. That opened this summer and has accrued very, very well at the practices. This is cluster randomized, and it's off to a very brisk start. So thanks to everybody who's participating in that study for really getting that out of the gate, rolling along. And then pushing hard to the finish line, we have another Alliance study, which is a breast cancer decision study. Dr. Heather Newman is the PI for that. There's 10 practices that are participating in that. And the hope is that that study will close to accrual sometime early in calendar year 2022. And then we have an ECOG Akron study 171CD, which is known as Smoke Free. That also is moving toward the finish line, looking at virtual tobacco cessation. So any of you who are participating on these 2 studies, we really encourage you to continue that hard work and get these moving. And then the NRG study that is looking at survivorship, care planning, and prostate cancer patients is doing a very nice job and is on track to close in the first quarter of 2022. So a lot of work getting these last few studies to the finish line here, and then again, multiple other protocols that are open and accruing. And I see in the chat that the exciting news is the credit study enrolled their first dyad today. That's kind of exciting. Thanks. That makes my talk a lot more exciting and easy. So it's great to the first group that put them on. Congratulations. Now, as we move forward, thinking about where we've been, we've heard some nice discussion here today. Joe Unger gave us a great presentation of kind of where we've been with accrual and how the COVID pandemic has affected accrual. And I wanted to show some of our CCDR accrual by the funding year. And as you know, our accrual is not just patients, it's also non-patient individuals and organizations that get enrolled on our studies. So the patients are depicted here in the red bars. The gray bars are the non-patient or individuals, and then the dark blue or black bar, depending on what's showing up for you, are the organizations or practices. And as you can see that in the funding year 2 years ago, that started in August of 2019 and closed in July of 2020, our numbers were higher than they are in our current year that just closed on July 31st of 2021. So certainly the pandemic had a role to play in that, but also the studies that we had open. We had some very large cancer care delivery studies in place, the OASIS study from Wake Forest, the TRACER study from SWOG. So that is why you see the numbers back 2 years ago up near 2,000 for the enrollment for patient accrual. Looking at the non-patient accrual, the individual, we had almost 800 2 years ago, and certainly the number is lower here in the current year, more in around the 250 range. And that again is because we had a couple of studies that had large numbers of clinicians that enrolled. And these were a study out of ECOG Akron as well as a study at the Wake Forest Research Base. And both of those have closed and are under analysis along the way. And then looking at organizations coming in, we still have a fair number of organizations going on because we've got newer studies coming up that are cluster randomized as well that is collecting data from the organization itself. So about 110 practices have contributed to organizational accrual over the past year. And that's a little bit less than 2 years ago, but really still doing very well. So while our numbers are down, some of it related to the pandemic and enrollments being down at the sites, others is related to just where the portfolio is. And now we've got, as you could see, some more studies opening up. On the next slide, you'll see the accrual that we have by minority. And we always keep an eye on this because we want to make sure that we are staying within what we are seeing for overall accrual. And we compare this to the numbers in cancer control and prevention as well. So about 20% of our accrual is minority accrual, which is consistent with where it's been in previous years. So the past year was 20% minority. I will say the one thing that we do have noted is our accrual of Hispanic populations is quite low. It's in the 3 to 4% range, so much, much lower than we hoped it would be. And so you are going to be hearing from NCI that we're really encouraging our research bases to have translated instruments if at all possible, and to look at the opportunity to enroll Spanish-speaking participants on their studies. So on the next slide, I wanted to talk about some of the upcoming opportunities we have for your team members and yourself to participate in some cancer care delivery related training. And we've got what we call a seminar series coming out this fall. It's entitled Collaborating to Develop New Practice Randomized Trials in NCORP for Cancer Care Delivery Research. And there's going to be 3 separate training sessions or seminars that will be held. And you can see here, the first one is November 9th from 4 to 5:30 p.m. Eastern time. The second one will be held on December 7th from 4 to 5:30. And then the last one will be held in January. We don't have the exact date on that yet. That date is still to be determined, but again, from 4 to 5:30. You do see here some target audience members for the different topics that are being presented. However, I will tell you, everyone is welcome to attend any session they're interested in. So if you have team members who might be interested in this topic and want to participate, please feel free to get them registered. And there'll be more information coming out through the NCORP listserv regarding registration details. And you can register for 1 session, 2, or all of them. It's really up to you. Another opportunity we have on this next slide is an upcoming series that we have a meeting coming this week here at NCI. It will be this Thursday, Friday, and next Monday, addressing social risk in cancer care delivery. And I think this is going to be an outstanding meeting, really focused on understanding social risk and how it affects our delivery of cancer care. There are going to be speakers from all over the United States participating in this meeting. And again, it's Thursday, Friday, and Monday of this week. You see the registration link there. I can go ahead and put that in the chat during this meeting as well. Registration is still open, and you can attend 1 day, 2 days, or all 3 days. You don't have to be able to attend all 3 to go. You could do your portions that are best for you. So please share this with your team members. We're really looking forward to this terrific meeting this week. And then as I finish out here on the next slide, wanted to bring up to all of you, because you always give me such great feedback at these sessions that we have. And this is where I really miss being in Chicago and being in the room with all of you. But for all of you who are ramping up to participate on the credit study, you know there's now a requirement that all of the affiliates or sub-affiliates complete a one-time practice level data collection form. It has 5 or 6 questions on it. Really should be pretty straightforward. And the goal to collecting this data is so that we can get an understanding of the heterogeneity and diversity of the practices and organizations that participate on our studies. But we have found that a couple of the questions are confusing or difficult for you all to do. So really would love to hear in chat if you could put in there, what is your, what really are your key things that we could do to improve this? The one question is here, this very first one about the practice setting. We have heard from some of you that it was misunderstood that when we used the term, it was a hospital or clinic that includes a health system that does include a health plan. That was interpreted as you as an employee were offered insurance, health insurance, which is not what the meaning for that was. The intent was that it was really, do you have a health plan like Kaiser has a health plan that an employer could purchase? And on the last slide, we realized that there was another question that asks about new analytic cancer cases. And some of you have that rolled up in your CTEP IDs, and others have it so that you can answer that individually. So would love to hear in chat if we could get some feedback here on these questions, what's helped you and what could be of benefit, because we really want to make this form something that's easy to use. Thanks so much.
Thank you so much, Kate. Appreciate that overview. And it looks like there's some really interesting sessions coming up. And I am assuming that people will receive invitations for that and their notifications about those. Great. All right. Well, we next have Allison Caban-Holt, SWOG's Recruitment and Retention Chair, who will get us up to date with some of the RRC activities to support our trials. Allison is the Associate Director of the Maya Angelou Center for Health Equity at the Wake Forest School of Medicine.
Hello, everyone. Can you hear me?
Yes, we can hear you, Allison.
Okay, great. Thanks for the introduction, Monica. So I promise that I will make this fast and furious, mostly fast, kind of hold back some of the fury. This is going to be just a quick update on the Recruitment and Retention Committee, as well as some diversity, equity, and inclusion efforts across SWOG that we are doing collaboratively. Sometimes I will refer to diversity, equity, and inclusion as DEI, so just wanted to let you all know that. It's okay if someone wants to advance the slide. I can tell if I was doing it or not. Okay. So our Recruitment and Retention Committee, our mission is to advise leadership with methods for recruiting and retaining minority patients on SWOG trials, as well as the recruitment and retention of minority SWOG members and leaders. We really feel like this is part of the guiding principles of SWOG that have been indicated in our SWOT analysis. In time, we plan for the Recruitment and Retention Committee to be added to protocol reviews so that trials being developed will have the opportunity to have to address recruitment and retention as at an early stage in development. But you can feel free to reach out to us for guidance and advice on recruitment and retention. Next slide. Thank you. The Recruitment and Retention Committee also has a diversity, inclusion, and belonging strategic working group that has a special focus in DEI. We are planning to develop guidance on DEI for study teams using existing evidence-based models and developing and supporting culturally responsive recruitment tools and outreach strategies. Next. Thank you. Another thing that we're doing in the RRC is our Take Action series, and we have looked at that as an opportunity to really share with study teams ways that they can reach out through actions, and not just through words, but also through actions, reach out to people of color and other underrepresented groups in SWOG trials. This has been conceived of by our education working group leads, Kamara Mertz Rivera and Lucy Gansauer. So back in April, we had a session that looked at ensuring health equity in SWOG among clinical SWOG trials among African Americans. And you see we had a fantastic panel. So what I want to let you know, next slide, is that tomorrow we will have another in our Take Action series that will be looking at ensuring health equity in SWOG trials among Asian Americans. We have Dr. Richard Lee. He's going to be our keynote speaker, and he has recently written a fantastic journal article in the Oncologist looking at disparities in cancer care and the Asian American population. We also have great panelists, including Naoto Ueno, Jared Akoba, and Jessica Huang. And what we're going to be doing is really discussing recruitment approaches and lessons learned by these panelists and how to recruit and work with Asian Americans in clinical trials. Dr. Huang is the chair of S1614 as PI of that trial, and we're specifically going to be looking at that trial and talking about strategies of an ongoing trial. So it'd be a great opportunity to join us in looking at how we can reach out to other populations. Next slide. So now I want to talk a little bit about some other DEI efforts. We have gotten generous funding from the Hope Foundation as well as Genentech in collaboration with the Patient Advocate Committee. And we are, one of the areas that we are using that funding for is to, we have a diversity, equity, and inclusion consultant, the Pope Consulting Group, and they are helping us develop DEI research and leadership training. So right now they have completed leadership interviews, have done anonymous focus groups, and we'll be using that feedback to tailor an inclusive leadership training that is going to begin next month. Next slide. We also have diversity, equity, inclusion champions. There will be 5 of those who will be working with study teams. Next slide. Our DEI fellow, Dr. Patricia Robinson, will be analyzing baseline data and will be hoping to increase enrollment of minorities and underserved participants by 20%. And she will also be helping to implement Team Science Module 6. Next slide. Team Science Module 6 will be available to everyone very soon. It's been shared with our RRC and our Patient Advocate Committee, and will soon be shared with leadership and study teams. Last slide. And that's my contact information. Please feel free to reach out to me if you have questions or want to learn more about the RRC. Thank you.
Thank you so much, Allison. I just want to also mention that the Take Action series is actually tomorrow at 12:30 Central Time, correct? And it's not noted in the agenda as Take Action. It's called the Retention and Recruitment Committee. So if anyone's interested, the entire time is going to be spent with those amazing panelists, the specialists. So, and also your Take Action series that was in the spring, that is available in SWOG.org under a group meeting under the April meeting. So that recording is available if anyone's very interested. That was a really great session. Thanks very much, Allison. All right, it's now my pleasure to turn the floor over to our team of clinical research data coordinators from Seattle, who many of you have interacted with on a regular basis. Diane Liggett will lead a panel discussion about data quality and navigating the maze of SWOG NCORP clinical trials.
Thank you, Monica, for the opportunity to speak. I'm Diane Liggett, as Monica said, and I am one of the SWOG NCORP clinical trial data coordinators in Seattle. Today, the other SWOG NCORP clinical trials data coordinators and I will discuss a few topics that will help you navigate through the maze of SWOG information to reach our mutual goal of quality data submission for the SWOG NCORP trials. You can contact all of us for protocol questions in all 5 SWOG cancer control committees by emailing cancercontrolquestion@crab.org. Next slide, please. I need all of you to share in the chat box your favorite strategies for quality data submission or any questions that you have for the data coordinators. We've been a little quiet today, so I want to hear from you in our session. As you are typing your chats, I would now like to introduce my first guide in the maze, which is Matt Ink. Matt, can you share some data submission resources and recommendations for screening and following study patients, which will get us on the right path?
Yeah, thanks, Diane. To answer your question, we'll first go over some important points to keep in mind when screening participants and evaluating eligibility. Going straight to the first point here, almost all SWOG trials will have their eligibility criteria outlined in section 5.0 of the protocol. So if you ever have any questions about eligibility, that's always the best place to look to first. In section 5.0, you'll see all the criteria that the patient must meet in order to participate in the trial. It's very important that each and every criteria is met because it is SWOG policy that no exceptions will be allowed to any of the eligibility criteria. The next point after that to keep in mind is that there may be certain study assessments that need to be completed prior to registration. These could include study-specific patient questionnaires, labs, or other assessments outside of standard care that may need to be completed prior to registration in order for the patient to be eligible. Finally, it's important to be aware that registrations cannot be canceled even if a mistake is made and an ineligible patient is registered. There is no way to reverse or change the enrollment. So it really does pay off to carefully review the eligibility criteria since there's essentially one chance to get everything right in terms of registering the patient. So if you're ever unsure about patient's eligibility or just have any questions at all, always feel free to ask us at cancercontrolquestion@crab.org, like Diane mentioned. If we can go to the next slide, please. So after you've successfully registered a patient, one of the next steps is probably figuring out what their visit schedule is going to look like. A general rule of thumb is that when calculating target dates for assessments or visits, the day of registration is day 0, meaning you should count from the day of registration when calculating the target dates, unless the protocol says otherwise. So you can typically find a very detailed data submission schedule in section 14 of the protocol, which should describe exactly how the study time points are going to work. So just make sure you're absolutely clear on how the scheduling will be determined on a study, because it can vary. And finally, date calculators can be very useful, especially when planning out dates far in the future, which can be a little difficult without a calculator. There's resources in the Cra Workbench, which we'll talk about a little more later on. And you can find a number of free date calculators online to help you as well. And that's all I have, so I'll pass it back to you. Thanks, Diane.
Okay, thank you, Matt, for sharing. My next question is for Kim Carvalho. Kim, can you please provide guidance on how sites can prevent or resolve some of the common issues that may happen in submitting data?
Yeah, thank you, Diane. Can we go to the next slide, please? Hey, okay. Once you get past screening and registering study participants, it's time to start submitting forms and biospecimens as required by the protocol. And with data submission comes the dreaded query and expectation reports. No matter how you track your queries and expectations, either by checking RAVE or by using the reports function on the Cra Workbench, you most likely will end up finding a query in RAVE that needs answering, or a biospecimen expectation that's showing is overdue in your report. I'm going to talk a little bit about how you can deal with the everyday errors that every site will experience. Next slide, please. RAVE queries come in several flavors, with the two most common being queries from the system and queries from the data manager. Queries marked open to cite from system are automatically generated when a required field is not completed or entered data conflicts with what is already in the SWOG database. For example, if you enter a last contact date prior to what is currently listed in the database, a query will pop up and inform you that you need to contact your data coordinator. Or if you missed entering a required measurement on the form, RAVE will remind you that the data field is required and needs to be completed. As a reminder, responding to the query in the query response box will not amend the form or update the SWOG database. You'll need to click on the pencil icon on the right-hand side of the page to edit enter data, or simply enter the required data in the data field if you left it blank. A rare but really annoying issue, phantom queries happen when users deactivate log lines or forms before a query has closed out by a data coordinator. Once deactivated by a site, SWOG staff are unable to close out the query. It no longer appears in RAVE, but continues to haunt your query report. To resolve this issue, review your query report to see which form the query appears on. Once you have located the form or log line, reactivate anything that has been deactivated and resave the log line or form. You will then need to contact your data coordinator to ensure that the query is addressed and closed out. Next slide, please. Next up, the specimen tracking system is an essential part of the data submission process for many protocols. It acts as a hub linking sites, SWOG, and the receiving laboratory. Logging and shipping specimens via SpecTrack will resolve the expectations for each patient and allow all parties to monitor which specimens have been submitted. We have recently expanded permissions to allow sites to update some data points for specimens that have already been submitted, such as date and time collected and site contact information. For all other categories not listed on the slide, you'll still need to contact your data coordinator or email technical questions at crab.org to request any changes. An important but often overlooked link in SpecTrack is the Notify Specimen cannot be submitted link, located in the same menu as the Log a Specimen link. Using this notify option will resolve expectations for biospecimens and also notify SWOG staff as to why a site was unable to ship the specimen. Queries and expectations are just two of the different ways that SWOG staff communicate with sites. Neither method is meant to punish sites, but instead provide ways in which we can all track the flow of data between sites and SWOG and ensure that there is an audit trail to track data changes. No matter what type of data submission issue you're having, remember that you can always contact us and reach out to your individual data coordinator for assistance. Thank you.
Kim, thank you. I'm sure your guidance will be very helpful in reducing the number of queries and unresolved expectations going forward. Our next guide in the maze is Sam Zingle. Sam, sometimes a patient is registered to study, but does not fit eligibility criteria, or the patient moves and cannot be located by the site. Can you please remind us what data should be submitted on these patients?
Sure thing,
Next slide,
Diane. Can you advance the slide? Okay, perfect. Thank you so much. Yes, so what Diane was talking about earlier, one potential obstacle in the data days, we so-called, can be clarity around the site's responsibility to patient follow-up. My colleague Matt had mentioned earlier that it is very important to verify all eligibility criteria before registering the patient in open, because once that patient is registered, they must be followed per treatment guidelines in the SWOG protocol they are enrolled to. It can't be canceled. They have taken up that slot. They need to be followed. SWOG policy number 30, item number 1, states that all institutional and individual participants in SWOG are responsible for the follow-up of all patients registered by the institution and/or the individual at the institution for as long as the patient remains alive, or for a protocol specified length of time. Follow-up requirement is defined as a minimum the last date the patient was known to be alive, or the date of contact, or the date of death. Or an optimum is the last date of the patient was known to be alive with a clear definition of disease status, including any second tumors or the date of death. So if you have a patient that has been registered to the study, they no longer want to be followed per protocol treatment guidelines, part of informed consent should include offering direct or indirect follow-up to the patient. Patients may not know that this form of follow-up may be an option. They may feel that they must agree to all aspects of the study or none of it at all, but that's not true. They do have options for follow-up, so please be sure to include that as part of the informed consent process. Remember that a patient is only considered a loss to follow-up when it's been at least 2 years of no contact and you have 3 documented telephone attempts and a certified letter to the patient that has been returned addressee unknown or no reply. Until both those conditions are met, those patients are still on the study. Item number 9 of SWOG policy 30 states that if a patient withdraws consent after registration, the institution must determine with the patient whether 1, they no longer wish to be treated per protocol, 2, they no longer wish to be followed per protocol, or 3, both. And if they meet that both category, then please complete the consent withdrawal form. In the off reason specify field, please state that the patient withdraws all consent to treatment and all forms of follow-up. Next slide, please. And then this next slide is just a screenshot of SWOG policy number 30. My presentation covered largely just items number 1 and number 9 that you see there. Roxanne is going to have more information on where we can find all these great SWOG policies and procedures here in a minute. But of course, with the situation with responsibility of patient follow-up, if it's getting really tricky and you guys are not able to get into contact with some of these patients and you're trying to figure out what data capture is going to look like in RAVE after that, just give your data coordinator a call or give us an email at Cancer Control Question, and we will try our best to help you out. Thank you so much, and I'm going to bring it over back to Diane.
Sam, thank you for sharing. My next question is for Roxanne Tupaccio. Roxanne, what are some of the other important reference materials that can help with submitting quality data?
Thanks, Diane. Well, no matter our role, valuable to any job really is simply knowing where we can find the resources and materials that can help us navigate our work. On the CTSU, staff access protocols, CRFs, and portals to upload regulatory and source documents. In addition, however, each of the network groups, including SWOG, post group-specific policies and resources to help sites navigate and manage their group's trials. In our final few minutes, we'll take a brief tour of the materials and resources specific to SWOG that you can find on swog.org. We'll start with the CRA Workbench, the Oncology Research Professional Manual, and best practices for SWOG studies. And next, we'll take a look at SWOG policies and procedures and make sure that you know how to reach out to us with questions. We'll start here on the SWOG homepage. Remember that while limited access is available on the site without signing in, using your CTEP credentials to log in will grant you full access to all content. Let's begin with the CRA Workbench. Next slide, please. The CRA Workbench is found on the homepage here under Member Resources. This is your primary dashboard for links and tools to manage your NCORP and NCTN clinical research trials. The Workbench has sections on patient management, such as links to RAVE and SWOG's specimen tracking system, along with pages on serious adverse event reporting and the application for planned unblinding. You will also find current and monthly expectation reports, the CRA newsletters, access to date counters and helpful formulas under tools of the trade, and documents on how to get started for the first time in iMetadata RAVE. If you're new to SWOG trials, or if you could use a refresher, the CRA Workbench is an excellent place to start. Next slide. Next up is the SWOG ORP Manual. ORP stands for Oncology Research Professional. If you consent patients to trials, ensure protocols are followed, oversee data safety and monitoring at a hospital, cancer center, or clinic, this manual is meant for you. There are multiple ways to access this on the homepage, but I've highlighted the two most common pathways here. Next slide. The ORP manual is a collaboration between SWOG SDMC staff, SWOG operations staff, with contributions from both the SWOG Pharmaceutical Sciences Committee and the ORP Committee. It has over 16 chapters and appendices that cover both administrative and data management issues and should be used at the sites as a primary reference tool in the day-to-day management of SWOG trials. Included are details on preparing for audits, registering patients, and understanding SWOG expectation reports. You'll also find instructions here on general form submission, study-specific form submission, response assessment, and you can review the differences between documenting adverse events on the study assessment forms versus reporting serious adverse events in the CTEP AIRS application. Next slide. The best practices for SWOG studies document is found on the homepage under Tools of the Trade in the CRA Workbench, and also under Quality Assurance and Audits, found under the Clinical Trials header. Developed by Elaine Armstrong, SWOG Quality Assurance Manager and her team, this provides information and best practices on protocol compliance, documentation practices, and consenting. It's meant to assist sites in avoiding deficiencies during a SWOG audit and offer standard procedures to follow in the absence of protocol-specified guidelines. Next slide. SWOG policies and procedures are found under the About section on the homepage, as shown here, and provide a high-level overview of SWOG's mission, constitution, and bylaws, and guidelines for membership. You will also find our data evaluation policy and procedure, ethical and regulatory considerations, as well as SWOG guidance on social media procedures. There are two policies here, however, related to patient follow-up that we reference often with our sites, and those are found here as well. Policy 12, SWOG registration and treatment policies, which Matt summarized in detail at the start of our panel, and Policy 30, responsibility for patient follow-up, reviewed by Sam, which details SWOG policy on managing patients who withdraw consent after registration to a study. Next slide. And finally, on the CRA Workbench, you'll find a Contact Us page with a directory of helpful email distribution lists. This is different from the Contact Us page on the main SWOG site. While our colleagues who manage the NCTN trials can be reached via one of the disease-specific email distribution lists linked here in the bright blue, All the data coordinators that you've just met share one email for all the trials under the 5 SWOG NCORP committees. That email, which you've heard often, is cancercontrolquestion@crab.org. Please remember to include both the study ID and subject ID in the subject line of your email, and be sure your content is free of all patient PHI. The primary data coordinator assigned to the study will respond. I'll end here, but we hope that this brief tour will encourage you to explore the resources available to you on swog.org, and that you'll find these helpful in our shared work. Diane?
Roxanne, thank you for that wonderful tour. That is, I think, one of the places a lot of the sites get stuck on is, where do you find this in the maze? So thank you for that. That was wonderful. I would now like to also thank, we have Julie and Mary. They put some suggestions or questions in the chat box. So I'd like to go to the chat box. And first we have Mary, and she has, our site runs reports each Monday for delinquent and data due. So that is a great strategy to get on those expectations in the queries and make sure your data is submitted on time and that we're getting responses. So thank you, Mary, for that. And then Julie says, we've had an issue with certified letters being undelivered, especially during COVID. Patients did not answer their doors to sign for the letter, or they did not go to the post office to pick up the letters. Many returned unsigned and undelivered. If you know that is something that's happened because of COVID. If anybody has any strategies to help Julie out there, please put them in the chat box, because I'm sure she'd really appreciate that. If I'm going to give you just a few seconds to go ahead and put some other questions in the chat. We really would like to hear from you at the sites. You are the ones that are on the front line there, and you know the best strategy. So please put it in the chat and share it with everyone, because you are very helpful with that. So next slide, please. Okay. In conclusion, I'd like to thank you for your attention and appreciate each of you for the work you do for the SWOG NCORP studies. If you have any additional questions or data submission strategies, again, we'd like to hear from you, and you can do that by emailing us at cancercontrolquestion@crab.org. And I very much appreciate, as I said, everything you do. Monica?
Thank you, Diane, and all the data coordinators for that, walking us through the data maze. And like Diane mentioned, if you have any additional questions, and all of our data coordinators mentioned, be sure to email cancercontrolquestion at crab.org, and we would love to even share some of your tips and tools with others. So right now, unfortunately, we have almost exhausted our 2 hours today. It's hard to believe. I know that usually when we're in person, we get to do a stand up and stretch. So I hope that you were able to do a little bit of that while you listen to our presentations. Next slide, please. So our next NCORP workshop is going to be next slide, please, Matt. Is in Chicago next year, October 19th, 2022. So our plan is to see you in person and be able to share information amongst ourselves. That's one part that we really miss, is our breakout sessions, where we have an opportunity to exchange ideas with each other, as well as with study chairs and other leadership. Next slide, please. We'd like to really thank each of you for joining us today, and a special thanks to our presenters and our panelists and to our meetings manager, who has helped us navigate the maze of these presentations. Please contact us about the workshop or any of the SWOG NCORP trials. We'd love to hear from you. And since this is the first day and maybe the first presentation that you have attended, the first session of the fall group meeting, we wish you well for the next few days, and hope to see you in the space of some of these NCORP meetings, and we wish everyone very well. Thank you so much, everybody.
Thank you, Monica.
Thank you.